Hello everyone, welcome. This is MEENU AGRAWAL. So, we will talk today about further about the structure of the plasma membrane. A quick review, let's do. We talked about cell membrane. We saw the composition of the cell membrane. It is made up of your lipids and proteins. And I talked a lot about the lipids, about the fluid mosaic model I told you about. Now today, basically, now we will talk more about proteins. Okay. So let's talk about proteins this time. Okay, so we know that these proteins that I talked about, there are two kind of proteins: your integral protein and your peripheral proteins, and your peripheral proteins, they were what, they can be easily separated as they are loosely bound, and integral protein are intrinsic part of the membrane. Okay, and we know that the proteins, they are now the membrane is highly dynamic structure, and the protein is not on the all the sides similar type of protein is not present, and they play different kind of roles. Like what they do, the function, they help in what, transport. They help in transporting water soluble substances. They help in transporting information. You know, they even transfer information. They act as receptors molecules, isn't it? They also act as enzymes. They have a role. Okay, so various roles or various functions are being done by these proteins, and many of them, they act as channels, gated channels. We will talk about them as they regulate the passage of the nutrients. They regulate, okay, they regulate the passage of the nutrients. Now, uh, okay, so let's look at the different kind of the now your this what are the different roles which is being played by them. Okay, so here in the diagram, as you can see here, what you can see, that they have got a various roles. I have written that these proteins, they do. Now what these proteins, they do, as you can see here, now they are acting as what, they are acting as an anchoring molecule. They help the, now the, like this molecule you can see here, this is the molecule which is integrin. Okay, and this is helping now the membrane to attach to the substratum. Okay, so you can see it is also helping to attach to the extracellular matrix, and also with the microfilaments that is within the cell. Okay, so they help a lot. Proteins have got, they have got diverse role, as I said now, ki every information that is there in your cell, it gets now expressed in the form of proteins. So these proteins have got various roles in your body. Okay, now they also act as your what, passive transport. We will talk about it in detail, ki how these substances are passively now transported from one part of the membrane to the other part of the membrane with the help of these proteins. They act as what, carrier molecules. Okay, and they also act as they also help in active transport. I hope you know about passive and active transport, at least this much, that passive transport is along the concentration gradient and it doesn't require energy, and active transport is against the concentration gradient and it requires energy. We will talk about it in detail about how it happens. Okay, so this is the role of, you can see now various proteins. Now let's look further what roles they have to play. And even with now we will see here, see this is acting as what, here your, this is acting as what, this is acting as your enzymatic activity. Now you know ki, now when you will read about respiration, photosynthesis, you will see ki how this protein, these enzymes, these proteins which are acting as enzymes, they are located on the membrane, and how various kinds of reactions are being conducted by them. So they catalyze various reactions as well. Okay, so this is the role of the enzyme, and even in they play a role in signal transduction. What is signal transduction? Signal transduction, like especially we see in case of your now various examples are there, like hormones are one of the best example I can give you here. Like in case of signal transduction, what is happening? Suppose there is a signal, okay, which is coming from outside. Okay, suppose this is a hormone. Okay, in this case the hormone is acting as a signal. It is binding to these receptor molecules. Okay, these are called as receptors which receive the stimuli, and they this no, transduce that information. Okay, so there are various mechanisms by which this information is further carried on, and ultimately what happens, the cells gives the response, the cell produces the response in now according to the stimulus given. Okay, so that is also that is called a signal transduction. Okay, and then another comes your cell recognition. Okay, so we have seen in the previous class that your glycoproteins and glycolipids, they were acting a very important role in this case of cell recognition. And there are proteins also. Okay, there are various antigens, okay, on a bacterial surface which is being recognized by these proteins. Okay, so you know that um, they have role in this cell to cell recognition. Okay, and then the last not the least is your intercellular junctions. You know that how does a cell communicates with now see this is a cell, okay, and this is another cell, third cell, fourth cell, there's so many cells. Now how do these cells they communicate among themselves? How do they communicate? So with the help of these intercellular junctions, there are different kinds of intercellular junctions, like there are tight junctions. Therefore, there are different kind of junctions. Okay, you will read about them in higher classes. So these junctions are basically what they do. They are like acting as a way of communicating. The one cell communicates with another cell, like you communicate with your neighbor. Similarly here also one cell is communicating with another cell with the help of these what junctions. So this is there with them. Just I'm telling you what role the proteins are playing. And now one of the major important thing you can see that now just let me make for you that the membrane has got what, it has got lipid. Okay, and these are the lipid molecules, phospholipid molecules I am making for you, and in between there is what, there is protein. Okay, now these are the lipid molecules. Okay, here also I can make. Now for you know the function of the plasma membrane, we call that the cell membrane or plasma membrane or cell membrane, in short I am writing. They are what we call them, that they are selectively सेमी परमिएबल. हम ये कहते हैं कि दे आर सेलेक्टिवली सेमी परमिएबल, है ना, दे आर सेलेक्टिव इन नेचर. क्या अंदर जाएगा, क्या बाहर निकलेगा, इसमें सेल्स क्या है, बहुत सेलेक्टिव रोल प्ले करते हैं. जाहिर सी बात है वो चीजें आराम से सेल के अंदर आ जाएंगी जो कि which can diffuse easily through the lipid layer. Okay, so they are basically your lipid soluble substances. Or you can say they are your non-polar substances. See, you have to understand the term polar and non-polar. Okay, so like carbon dioxide, oxygen, we can say here carbon dioxide, oxygen, now they can also diffuse very easily through this lipid layer. But your polar molecules, okay, your polar molecules, polar means what? Polar, the word itself tells you that it has got polarity. Means you know that the molecules, they some molecules are more negatively charged, some are more positively charged. Now if the molecule is like the bond is not towards one side more, okay, whether negative side or positive side, we say they have got a polarity. So such molecules are polar molecules. Now these polar molecules, like suppose I take ions, okay, so they are positively charged or negatively charged ions, so they are polar molecules. Now these molecules, how will they pass? They can't pass through these lipid soluble substances. No, they are not lipid soluble, so they can't pass through this lipid membrane. Okay, these are non-polar in nature. And even these protein that you see, they here you see the kind of the amino acids will be here, they will be non polar in nature, they will be hydrophobic in nature, and the ones which will be inside here, they will be hydrophilic in nature, because they are facing this hydrophobic part of the lipid. Okay, so what do we find, that how will these water soluble substances will pass, how will these polar ions will pass? They will pass through these proteins. Okay, so water soluble substances and your proteins can pass through it. For remember that now there are various channels, like one is your aquaporins. Okay, it's a kind of a channel through which water can pass, and it is present from bacteria till humans. Okay, and basically they are they are gated channels. Okay, it's not that they are open every time. Now see, some amount of water can move through, now water can move through it, through your lipid layer. When these molecules, they when they move, they create a space in between, and in that the water can pass through it. Okay, so through aquaporins also there is a huge amount of water that can pass. Okay, and these aquaporins are present from bacteria till now humans, they are present, you can find them. Okay, so all of them they have, and it is basically now helping in preserving water. Okay, so aquaporin is there, and it's a I told you that is a gated channel. Okay, so this is the basic thing which you need to remember. Okay, lipid soluble substances can pass easily through your lipid layer, because and your water soluble substances and your polar molecules, they pass through your this protein part, and therefore we say that plasma membrane or cell membrane is selectively semi permeable. It allows certain substances to move inside the membrane, and allows certain, definitely what it will allow to move, nutrients, food, oxygen, and those waste products, it will allow to move out of the cell. Okay, so nutrients, you can say your oxygen, यहाँ पे आपको waste and all, they move out of the cell. Okay, so now let's move on to what is diffusion. Okay, let's see what is diffusion. I hope you people know about what is diffusion. Okay, now diffusion is what? That we know that the movement of substances now from their higher concentration uh, to the lower concentration, or you can even say randomly these molecules they move, and this intermixing of molecules is called as diffusion. Like suppose I draw a beaker for you, okay, and I have a tube of sugar or potassium permanganate, anything it could be, and suppose this is water. Then what will happen? It will later on, it will diffuse into it. It will start diffusing into it, and you will find that after some time it has totally mixed. It has got mixed totally. Now it has got mixed totally into it. Okay, so this is called, now this is what we say, the movement of molecules. We say the movement of molecules And remember, this movement of molecules is quite random. Okay, so movement of molecules from its higher concentration to its lower concentration is called diffusion. ना देखिए, यहाँ पे आपको ध्यान देने की ज़रूरत क्या है कि जहाँ पे हायर कंसंट्रेशन है, तो जब ये हायर कंसंट्रेटेड है तो क्या है, this higher concentration is creating what? It is creating a kind of a potential gradient, है ना? Okay, so first try to understand what is the term gradient means. Okay, let me tell you what is the gradient. When I'm saying what that molecules move from its higher concentration to its lower concentration, huh? Or we can also say that the molecules move along their concentration gradient. Okay, so along the concentration gradient also we say concentration gradient. So first you need to understand what is gradient. See, gradient is what? When there is something at a higher position and something at the lower position, okay? Or something as more at some place and something is less at some place. So there is, what do you find? It is called as what? It is called as gradient. This is called as what? This is called as gradient. Okay, so this creates a kind of a gradient. Okay, so due to position it could be due to concentration, so it is creating a kind of a from higher to low kind of thing. So it could be at a higher position, है ना, जैसे पानी हम ऊपर टंकी पे होता है तो it's at higher level and water at lower level is there, or here more concentrated the solution is and here the less concentrated is, so automatically the flow of the substances from higher to lower. Okay, so this is called as concentration gradient. Why is it? Because due to their position, this is what? This is the potential energy they have. This is your potential energy. So using that energy, they are moving from one place to another. Okay, so therefore they here this is called as diffusion. This is simple diffusion we call it. Okay, this oxygen like moving in your lungs to your blood, and from your body carbon dioxide moving into your blood, it is all happening due to concentration gradient. So this gradient is allowing, the stored energy is your potential energy, okay, which has the capacity to do work as a result of position. Okay, so that is what is it, okay, which is causing the movement of the molecules. This is what we call as simple way of diffusion. Now there is another thing along diffusion which you need to remember, that is your osmosis. Now let's talk about osmosis. Now if what is osmosis? Osmosis is also movement of water molecules. This is also what? Movement. It is movement of water molecules. Across a semi-permeable membrane. Across a semi-permeable membrane. Along its concentration gradient. Or if I have to simplify it, I can say that movement of water from dilute solution to concentrated solution. I hope you understand, because dilute solution has got what? More water. The water is going to move. So from where water is moving? From where water is more to where water is less. Isn't it? I hope you should get it. So it is moving, the movement. See, osmosis is what? It's osmosis is a type of diffusion where there is only movement of water molecules happening, okay, across the semi-permeable membrane along its concentration of what we are going to talk about. We are talking about the concentration of water. So from where water is more to where water is less, it will move. Okay, so you must have heard about the term osmotic pressure. Okay, so let's talk about this osmotic pressure. Suppose I have this U-tube. Okay, I have this U-tube, and here it is separated by a semi-permeable membrane. Or okay, next slide. Now suppose I take this U-tube. Okay, now this is your U-tube. Now in between there is a semi-permeable membrane. This is what is your semi-permeable membrane. Semi-permeable membrane. Okay, now this is your solution which is is concentrated. Okay, suppose this is more concentrated solution. Okay, or you can say this is a solution having solutes in it. Okay, so this is solution, and here is only water. So what will happen? Definitely water is going to move from its higher concentration to lower concentration, and the water will start moving into it. Now if I don't want further water to move inside this solution, what I have to do? What can I do? I can, suppose solution is still here and water is, suppose I made with blue this water, okay, and this water is still here. Okay, now if this solution is still here, okay, now the water will start moving and the solution will start moving up. Now what I have to do? I have to stop from moving in that. Then in that case I have to apply a pressure here. So what I do, I take a piston and I just try to apply a force here with a pressure. I exert a pressure here, so that, and at that pressure where the further movement of water, see, there will be always, when it will come into equilibrium, what will happen? There will be no net movement of water molecule, but there will be constant exchange of water molecule. So the amount of water molecule moving here will become equal to here. So at this time, when it will come into equilibrium, that pressure is called as what? That pressure which doesn't allows further movement of water into the solution is called as osmotic pressure. That is called as osmotic pressure. Okay, so that pressure which won't allow further movement of water into it is called as osmotic pressure. Okay, now let us talk about now the type of solutions and see what happens. What are the different type of solutions? You must have read about it earlier also, so you know that this solution, if I'm talking about, suppose I make I take 3 beakers. Okay, in all of the beakers I have added what? I've taken 100 ml of water. I have taken what? 100 ml of water. Some may 100 ml of water. Okay, all of them have got 100 ml of water. Okay, now what varies in them is your, suppose in this I have added 1 gram sugar. In this I have added 2 gram sugar. Okay, and in this I'm not adding right now anything. Okay, now 1 gram sugar and 2 gram sugar. Which solution will be more concentrated? This 1 gram or 2 gram? Fine. So definitely you will say ki this will be more concentrated, isn't it? Why it will be more concentrated? I can ask you. Because the water is 100 ml on both the glasses. Now if I keep these two solutions across the semi-permeable membrane, will water move? Yes, it will move. Why? I tell you. Why this is more concentrated. Although the water is amount is same, but the solute, it is the solute which is very important here. Why? See, here the sugar molecule, I will just make a bigger one for you to understand it. Now, if I make a sugar molecule here, okay, and I make another beaker, and 2 grams, so definitely more solute will be there. Now, the amount of water that is available per solute molecule is more here, isn't it? But the amount of water present per solute molecule here is less. Okay, therefore, this is more dilute solution than this. This is concentrated in comparison to this. This is dilute. Okay, so what we call those solutions, and I can have a solution whose concentration can be same as well. Okay, so those, now what we call these solutions, let us see. The solutions which are having, now we use the word tonicity. Tonicity means strength of solution. That is called as tonicity. Okay, now the solution can be isotonic, it could be hypotonic, it could be hypertonic. Okay, isotonic means same concentration. The two solutions, and remember these are relative terms. We always talk about them in relation to some other solution. Okay, so it could be same concentrated, the two solutions could be. If I say it's hypotonic, means it is what? It is less concentrated. Less concentrated means it's a dilute solution, I can say. Okay, and hypertonic means more. Hyper means more concentrated, or I can say it is what? A concentrated solution, a conc solution, isn't it? A concentrated solution. More solute it has got less solute. Okay, so this is your tonicity of the solution. Now what will happen if I keep a cell under these different kind of solutions? If I keep, suppose I am taking a plant cell as well as also an animal cell. Okay, so with animal cell, suppose the blood is one of the best example, and with blood cells you can do this experiment also very well. Suppose I keep this blood cell, I'm just drawing for you. If I'm keeping this blood cell under your isotonic solution, under your, this is your isotonic, and then I am keeping your blood cell under your this hypotonic solution, and then I'm keeping it under now hypertonic solution. Then in each case what is going to happen? Here what will happen? Since it is an isotonic, means the concentration inside the cell, the concentration inside the cell and outside the cell is same. So net movement, there it's not that there won't be any movement of water molecule. There will be movement, but net movement will be 0. So just water molecules will exchange. Equal number will go inside and equal number will come outside. Okay. In hypotonic solution, where is water more? This is hypotonic, means this is dilute. Here water is more. The water is more here. And so water will move from here to here because it is more here. So water will move, and what will happen to the cell? It will swell. The cell will swell. If you keep it for a longer time there, okay, then it will burst. If you keep it for a longer time, then it will burst. Okay, and your hypertonic solution, this is hyper means less water. Less water means from cells, inside the cells water will come outside, and therefore it will what happen? It will shrink. Okay. Fine. The cells will shrink. Now, even if I keep a plant cell in this case, same thing is going to happen. But there will be a slight change in a plant cell when you keep them under hypotonic solution. Now, I'm just making for you, for this case, the plant cell. Suppose this is your plant cell. Okay. Now, I'm keeping this cell under hypotonic solution. So, it will swell. है ना, एकदम स्वेल कर जाएगा और ये एकदम मेम्ब्रेन के एकदम वॉल के एकदम पास में मेम्ब्रेन चला जाएगा, isn't it? And then the nucleus and all will be there. Okay, तो ऐसा दिखेगा ना, स्वेल कर जाएगा. जैसे one of the best example that you can quote from your day-to-day life, if you soak gram, it swells. Okay, पर अगर आप उसको ना, आप प्लांट सेल के केस में देखेंगे कि आप उसको ज्यादा देर भी छोड़ देंगे पानी में, okay, then it won't burst. It won't burst. Why it won't burst? Because see, it has got a wall and now it is a membrane which is very closely oppressed to it. Now when inside this membrane, if the content is increasing, the water is increasing, then what is happening? Then what is happening inside the membrane? This membrane is putting, this membrane is putting pressure. This membrane is putting pressure on the wall, isn't it? This membrane is putting pressure on the wall, and in turn, what is happening? And in turn, your wall is putting pressure on the membrane. Wall भी membrane पे pressure डाल ले. Okay? So do you remember osmotic pressure? I told you. हाँ? तो osmotic pressure किसका ज़्यादा होता है ना? I will talk about it that also. So you will see कि it will also act on a pressure on the membrane which will prevent further entry of water molecules in it. Okay, so they don't get burst. Okay, and if you keep the cell under hypertonic solution, okay, if I keep the cell under hypertonic solution, then what is going to happen to the plant cell? What is going to happen to the plant cell? It will shrink. Okay, it will shrink, and you can clearly see it under the microscope. Okay, and these cells are called as what? They are called as plasmolized. Plasmolized cells. Okay, because with reference to cell wall we are talking to it. There is no reference in case of an animal cell. Okay, so this term is not applied for your animal cell. It is only applied for your plant cells. So these cells are what? Now they have shrinked, and this cell is called as plasmolized cell. Again, if you keep these plasmolized cell underwater, they will again revert back to their original condition. Okay, so you can keep various substances under concentrated solution and you see them, what happens. So here remember one thing, key solution which is hypertonic, they have got a high osmotic pressure. They have high osmotic pressure. And solution which are hypotonic, they have low osmotic pressure. Okay, fine, because less pressure will be applied in this case. Okay, more pressure will be applied in this case to prevent entry of water, है ना? So that's why they have high osmotic pressure and they have low osmotic pressure. And I hope this is also clear कि why cell wall is not allowing, it's not causing the burst. Okay. Okay, and when you keep them in hypertonic solution, they get shrinked, and this condition in case of plant cell we call as plasmolysis. So today we have covered diffusion and osmosis. Okay, now in the next class, next video, I will talk about passive transport and active transport, and if possible about bulk transport. Okay, with this I will end and say goodbye to all of you, and God bless you.